Hello, my name is Isabel Emanuel, and I am a PhD candidate in the Department of Plant Pathology at The Ohio State University. And today I'm going to be talking about some of my recent work in the Ornamental Pathology Lab under the advisement of Dr. Francesca Hand, who is my co-author for this poster. I want to first quickly thank One Health for letting me present here today and then dive right into my research content. For the past almost 3 years now, I've been working on understanding the factors associated with an emerging fungal disease causing latent fruit rot in deciduous holly. This disease arose in the northeastern and central United States around 2009, and the associated symptoms are fruits which never fully matured, so they lack that desirable deep red color, and they also tend to be smaller and develop necrosis starting around the stigma end of the fruit. Here I have quickly pictured some healthy deciduous holly fruit juxtaposed with some of the symptoms that we typically see in these diseased fruit. So my advisor's lab started working on this pathosystem in 2015, and what they found was that the disease was caused by a complex of 7 fungal pathogens, and all 7 of these pathogens were able to reproduce disease in healthy, wounded, mature fruit. However, one of these fungi was able to also reproduce symptoms of disease later in the season when inoculated in late spring on open flowers. This delay in symptoms development suggests a latent infection, and the fungus able to do this is a newly emerged species of Diaporthy called Diaporthy illichicola. Now, I want to take a second to emphasize that we do not know if the other fungi are also able to cause this latent infection. We just haven't been able to reproduce disease this way with the other pathogens in the complex. So my work has focused on understanding the latency of symptoms development, and I've done a lot of work specifically with Diaporthy illichicola. This poster is showcasing a set of experiments I actually just completed, which investigate the strong negative correlation between temperature and disease incidence that we see in the field. As I mentioned before, the symptoms of this disease develop months after inoculation with Diaporthy illichicola and line up well with a seasonal field temperature drop that we see as we progress into October and November months here in Ohio. And this correlation made us really curious about whether the temperature was affecting the growth of these fungi within the complex directly, which is why we see this rapid symptom development later in the season. So to test this hypothesis, I selected 6 representative isolates of the 4 major pathogens in the fruit rot complex: Diaporthy illichicola, Alternaria alternata, Colototricum fiorinae, and Epicoccum nigrum. And I grew these isolates at temperatures ranging from 5 to 35 degrees Celsius and monitored the rate of spore germination and hyphal growth. So after collecting measurements of hyphal growth over 3 weeks and the percent spore germination, which occurred at each of these temperature treatments within 24 hours, I calculated the area under the growth rate curves, and this is the data that you can see in the results section of my poster. I separated the means here in R using Tukey's HSD with an alpha value of 0.05. So what we're seeing here is that Diaporthy and Alternaria both prefer temperatures above 15 degrees Celsius. Colototricum fiorinae, on the other hand, can still grow optimally at temperatures as low as 15 degrees Celsius, and Epicoccum nigrum is able to grow and germinate optimally at 10 degrees Celsius. All of these pathogens, however, were significantly decreased in their ability to germinate and grow at 5 degrees Celsius. So, based on these results, there is a possibility that Epicoccum could take advantage of temperature drops. However, given the previous research, which showed that Diaporthy illichicola and Alternaria alternata are the 2 most frequently isolated pathogens from diseased fruit lesions, it is unlikely that temperature alone is responsible for the late season symptoms development given the lack of these 2 fungi to grow well below 15 degrees Celsius. So all in all, these experiments successfully disproved the hypothesis that cold temperature directly affects fungal growth in a way which leads to symptoms development in late season fruit rot. And our future work will continue to explore potential factors associated with the latency of symptoms development. These include increased sugar concentrations as fruits mature, increased light exposure to the fruit as the leaves drop, and decreased defense compounds within fruit during ripening. And ultimately, understanding the system will allow for us to create an integrated disease management plan to sustainably reduce crop loss. And with that, I conclude the content of my poster presentation. I'd like to thank my funding sources, the CFAES Seeds Grant Award Committee, the TJ Kavanaugh Foundation, and the USDA NIFA Hatch Project. And thank you everyone who listened today. If you have any questions, please feel free to email me or visit our lab website at u.osu.edu/handlab. Thank you.